Dr. Andrew Pieper has dedicated his career to advancing the understanding of the molecular processes that cause neurodegeneration and neuropsychiatric disease, and to discovering and developing new treatments for patients suffering from these devastating conditions. He currently serves as Professor of Neuroscience and Psychiatry at Case Western Reserve University and is Director of the Case Western Reserve University Translational Therapeutics Core for the Cleveland Alzheimer's Disease Research Center. He is also Morley Mather Chair of Neuropsychiatry and Director of the Neurotherapeutic Center of the Harrington Discovery Institute of University Hospitals of Cleveland, as well as a member of the Geriatric Research Center at the Louis Stokes VA Medical Center of Cleveland. At the VA, Dr. Pieper has a weekly outpatient clinic in the psychiatry department that complements his focus on cognitive and mental health aspects of neurodegenerative disease and brain injury. Dr. Pieper earned his bachelor's in biology and chemistry from Earlham College and his MD and PhD degrees from Johns Hopkins University School of Medicine in Baltimore. He conducted his PhD thesis work in neuroscience and his medicine internship and psychiatry residency at Johns Hopkins Hospital. Dr. Pieper holds over 24 issued patents for his work. So with that, I'll turn it over to you, Dr. Pieper.
Okay, well, thank you, Anne, for that nice introduction. And I just want to say hello to everyone and how thrilled I am to have this opportunity to talk to you about something that's very important to me, which is the progress that modern science is making today to understand how the brain works and identify new ways that we can protect the brain and thereby protect ourselves from brain disease. My research lab is centered at these three institutions. And let's see, how do I advance the slide? Oh, there we go. Okay. These three institutions that are shown here, as Anne just said, Case Western Reserve University, the Louis Stokes VA Medical Center, and then the University Hospital Systems. And these are three independent yet very tightly collaborative institutions here in Cleveland. And what I'm going to talk about today are two things. First, work that we're doing in my lab across these three institutions to promote brain health. And then second, a unique enterprise that we have here in Cleveland designed specifically to foster worldwide development of new medicines for neuropsychiatric disease. So before I start, I want to define what neuropsychiatric disease means. And essentially, it means any form of an illness that has components of both psychiatry and neurology. Traditionally, neurology is thought of as the brain disease. Examples would be stroke or seizures or brain tumors, whereas psychiatry is thought of as diseases of the mind, such as emotional disorders or psychosis. But what I think a lot of people don't know is that this distinction was not always so clear-cut. And actually, up until the 1900s, the mid-1900s, they were pretty much in the same field. And that's because around the mid-1900s, we saw the onset of all different forms of psychotherapy, very structured psychotherapies that really made a tremendous difference in helping patients. Unfortunately, somewhere along the line, there became propagated this erroneous belief amongst a lot of medical professionals and the public at large that psychotherapy is working by processes that are independent of the basic functioning of the brain. And now we actually know that that's not the case. So, for example, modern imaging techniques have shown us that the process of psychotherapy induces many of the same changes in the brain that we see when we give people medicines, such as improving blood flow to certain regions or increasing the strength of different networks in the brain that allow us to cope with stress and function normally. And so we really need, I think, to come back together now with an approach to understanding how neuropsychiatric disorders emerge from common processes in the brain. Given all that science is capable of today, it's more important than ever that we move fully beyond this artificial distinction between mind and brain. And this really hit home for me personally several years ago when I was attending on the inpatient unit in psychiatry. And we received a patient from the medicine floor who had been transferred because he presented initially with mysterious accumulation of bruises on his face. His wife had brought him in because these bruises had been accumulating over the past several weeks. She couldn't figure out why. He said he was falling, but she didn't ever see him fall. So when he explained to the medical team what was going on, they said, well, your wife didn't see you fall, and we don't believe you that you're falling. And he became enraged and disruptive. And so the next thing they did was call the psychiatry consult service, who then evaluated him and decided that he would benefit from an inpatient psychiatric stay. So the consulting psychiatrist sent him up to the inpatient psychiatry floor with the plan to rule out as the possible causes of his bruises. Number one, intentional injury. Number two, an unintended consequence of surreptitious drug or alcohol abuse. And number three was possible psychosis, commanding him to do things that were not safe. The problem was that nobody had actually watched him walk. And so when we did our full exam on him, once he was on our floor, we noticed that he was ever so slightly shuffling his feet. And this means that he wasn't lifting his feet up enough as he should have been when he was walking. And this is a common cause of falling. And it's also an early sign of someone who might have Parkinson's disease. So we ordered a DOPA scan, which is a very advanced imaging technique to take a picture of the brain that deteriorates when people develop Parkinson's disease. And the next day, the results came back that sure enough, he had signs that were compatible with very early Parkinson's disease. So then we called the neurology service and they started him on the appropriate medication. His symptoms improved and everybody went home. And I say this because the point being that if as psychiatrists on the psychiatry floor, we had not been thinking about disorders of the brain that are outside the typical realm of psychiatry today, his diagnosis would have been missed and he would have continued to suffer these consequences and so forth. And so it is no longer sustainable for neurology and psychiatry to consider themselves as fully separate disciplines. The brain and the mind are one, and we can't afford to treat them anymore as separate entities because they both share a common root in neuroscience. And so I have endeavored to focus my own research on brain health accordingly. And it's very striking to me that my slide advancement is not working. Okay, thank you. It's very striking to me that the diseases listed here on the left, traditionally considered in the realm of neurology, and the diseases listed on the right, traditionally considered in the realm of psychiatry, all have a common finding of neurodegeneration. It's to different degrees, but all these diseases that I've listed here show death of nerve cells as the diseases progress. Neurodegeneration simply means loss of structure and function of nerve cells, which compromises our ability to function normally and eventually can lead to nerve cell death. And when this happens in the brain, it can be very devastating because the brain controls essentially who we are. And so as we experience neurodegeneration in the brain, we can have emotional changes, we can have personality changes, we can have extreme difficulty with our learning and our memory. And in fact, in the most extreme cases of neurodegenerative disease, people actually can forget even who they are. And so we need to do something about this problem. And I want to impress upon everyone just how tough this problem is today. And so if you could advance to the next slide. Right now, there are almost 50 million people worldwide suffering from neurodegenerative disease. And there are also no medicines available to patients to stop the process of nerve cell death. So in other words, for all these diseases, we have medicines that can treat the symptoms for a while, but we don't have any ability to actually stop the process of nerve cell death that is driving the disease to get worse and worse and worse over time. And if we don't do something about this, the problem is going to triple in the next 30 years. If you could advance to the next slide. So by 2050, it's projected that we'll have upwards of 130 million people with the disease. If you could go back one slide. And in the United States alone, this is going to cost upwards of over $1 trillion a year. So really the time is now that we need to do something about this problem that we're facing. And so we need to ask ourselves, why haven't the traditional drug discovery techniques worked for neurodegeneration? And what can we do differently? So as a very brief overview, the way that drugs have, medicines have been traditionally discovered for the field, for medicine as a whole, is that scientists have examined a problem in the body. They do their best to reduce it to just a few chemicals that they can then put into a test tube or a petri dish. And then they have a chemical reaction that they say represents the disease process in the body. What they do then is we take large robots and we screen millions and millions of compounds against those chemicals. And we try to find ones that make the chemical reaction move in a favorable way. And this has been a really good idea for medicine as a whole. And in fact, it's the reason it's worked spectacularly well for most fields of medicine. For example, cardiovascular disease. People are living longer, and a lot of diseases have been had new treatments developed, and in some cases cured, but it hasn't worked in neurodegenerative disease. And so people are living longer, and we don't have anything to stop neurodegeneration. So as a consequence, the prevalence of neurodegenerative disease is increasing even more. And so people are living longer, but they're not necessarily living better. And in many cases, they're actually living worse. And I think part of the reason is that the brain is so much more complicated than the rest of the body. And we haven't necessarily always, what works in other parts of the body to discover drugs has not worked in the brain as well as we would like. So when I began my first job as an assistant professor, I tried to take a different approach to this problem. If you could advance to the next slide. Thanks. What I wanted to do was I wanted to focus on seeing if we could make the brain itself into the test tube. And so I focused on a region of the brain in mice that is called the hippocampus. It's all mammals, including humans, have it. And I picked the hippocampus for two different reasons. First, it's one of the most important regions of the brain that controls our emotions, controls our learning and our memory, and controls how we adapt to a changing environment. And it's one of the major regions of the brain that deteriorates early and rapidly in almost all forms of neuropsychiatric disease. So if we could find a way to make the hippocampus function better, we could potentially impact a lot of different types of brain disease. The second reason why I chose this was because I specifically wanted to look at nerve cell death. And what's unusual about the hippocampus is that it's one of just two regions of the brain where we make new nerve cells every day of our lives. So there's a constant turnover of nerve cells in the hippocampus. And this is important because it allows the hippocampus to change as we age, so that we continue to adapt our brain to different environmental challenges and stresses. And it does this by having stem cells at its base that continually make many more nerve cells than we actually need. It makes an excess of them. And the ones that it doesn't need die. And so what I thought was, wow, maybe the hippocampus itself could serve as a test tube, in that we have a little microcosm of nerve cell death going on in the brain. And so what I did was simply take a few thousand different small drug-like molecules with no known previous function, and I infused them directly into the circulatory system of mice in the brain. And then a week later, I looked at their brains to see if any of those molecules had made the nerve cells survive at a higher rate. And after about 2 years, I found one molecule that did this, and it did it very effectively. And so if you just hit the keyboard once, it'll advance to show that molecule. Thanks. It's shown right here. It's called P7C3 because that was how I kept track of the compounds. And this particular one was pool number 7, compound number 3. And what I found was that when we gave it to mice, it doubled the survival of nerve cells in the hippocampus. So the next thing we did was say, wow, let's see if it actually works in disease models. And so we tried it in what I call the big 3 of neurodegeneration: traumatic brain injury, Parkinson's disease, and Alzheimer's disease. If you could go to the next slide. And so we gave it both orally or by injection to mouse and rat models of different types of traumatic brain injury, of different forms of Parkinson's disease, and a couple different genetic models of Alzheimer's disease. And we saw that it was very successful in protecting nerve cells in the brains of those animals from dying, and that correlated with preservation of the ability of those animals to function normally in terms of learning and memory and motor behavior, et cetera. So then we shared it widely with people all over the world. And if you just hit the keyboard one time, thanks. And we've now seen through our lab and in collaboration with others, and others have independently shown, that it also works very well to preserve nerve cell survival and function in animal models of stroke, of peripheral neuropathy, of spinal cord trauma, of Lou Gehrig's disease, also known as amyotrophic lateral sclerosis. And then importantly to me as a psychiatrist, by virtue of its ability to enhance hippocampal function, we've shown that it works well in animal models of depression and anxiety, as well as in facilitating recovery from a stressful situation, which is considered to be a model of post-traumatic stress disorder in mice. And so we were really surprised, actually, that it worked in such a wide variety of different conditions. And we didn't expect that at all. And it really finally made a lot of sense as to why it was working this way when we figured out how it was that the molecule worked. And so it works in a way that we never would have predicted or screened for before in a test tube. It works, P7C3 works by helping the nerve cells generate energy for themselves. So it specifically increases the availability of important chemicals that nerve cells need in order to generate enough energy. And the nerve cell uses this energy to fight off disease and fight off injury. An analogy that I like to use is that of an automobile. So most cars today still use gas, and the car needs gas in order to drive. When the car tries to drive faster, or if you have to make the car heavier, or if you have to drive the car up a hill, it uses more gas. When it uses up all its gas, it dies. And the same principle applies to nerve cells and energy. They use energy for all of their routine processes, but then they also use energy to repair themselves when they're injured and to fend off the genetic burden of disease. And when they run out of energy, the nerve cell dies, just how the car dies when it runs out of gasoline. And so we now understand how it works. And the next question is, can we possibly one day get this into patients? And that brings me to why I came to Cleveland, actually. So in addition, of course, to the world-renowned academic environment of Case Western Reserve University, I was also very attracted to the opportunity to join the Harrington Discovery Institute at university hospitals. And so while the Harrington Discovery Institute is pretty well known in academic circles, it's still pretty new. It's only about 7 years old now. And I don't think that many people in the community know about it. And so the next slide here is a diagram, a little cartoon that I've made just to give people an idea about what it is that we're doing at HDI. So it was founded about 7 years ago by an extraordinarily generous philanthropic gift from Ron and Nancy Harrington and their family in Cleveland. And their vision was that they wanted to find a way to help bridge this gap between basic science discoveries and medicines that get into patients. And the problem is that this gap is getting wider and wider, and especially in the field of neurodegenerative disease or mental health. There is a huge barrier towards getting basic science discoveries into patients. And as a consequence, a lot of discoveries are being made in labs and aren't being appropriately pursued and developed for their potential to make a medicine. And that's because we're not trained as scientists or doctors or physicians. We don't learn in medical school or graduate school how to do the things that are in this gap, so to speak. So this includes things like how to optimize your molecule to make it safer for people and more effective, how to do various chemical reactions to make it more stable and formulate it for delivery in an optimal way for a medicine, how to determine its various potential toxicities that might make it, might provide a barrier to drug development. And it also includes very practical things like intellectual property protection for new materials so that you can properly package it for commercial development, and also items such as how do you appropriately package all of your basic science results into a compelling application to the Food and Drug Administration to get approval for an investigational new drug? What are the strategies for that? And so what we do is we select scientists from, currently, we're hoping to expand eventually worldwide. Currently, we operate within the United States, the United Kingdom, and Canada. We select scientists with very promising initial scientific discoveries that we believe can get across this gap if we can help them do that. And then we surround them with a mini pharmaceutical company of leading scientists, Nobel laureates on our committee, retired leaders from the pharmaceutical industry. Everybody, we have a team of people who has vast expertise in all of the experts to get across this gap. And so if you hit the keyboard once, you'll see that we are attempting to bridge this gap at the Harrington Discovery Institute, and then hit it again so that the scientists can get across the gap and we can bring medicines to patients that would otherwise not be developed. The next slide shows the reach that we have so far. I've denoted here all of the projects that HDI is funding and helping, and then the ones in green are specifically neurotherapeutics projects. And I want to point out that since the inception of HDI, we now have 8 novel medicines in clinical trials. We have formed 27 new companies, and we've licensed 7 programs to big pharmaceutical companies. And so traditionally, the process of drug development can take anywhere between 15 and 20 years. But what we are able to do is take very promising products and have very focused no-go, no-go criteria, very focused milestone-driven achievement, so that we believe that and so far we are, able to shorten this process by about half, which we're hoping to apply to the field of neurotherapeutics in order to accelerate the development of medicines that are really, really sorely needed right now. And then the next slide is simply a list of the institutions that we're currently housing, that currently house the various investigators that we're supporting. Again, the ones in green are noted as the neurotherapeutics projects. And then the next slide is my final slide, which is just that I want to talk a little bit about the importance of the synergy of federal and philanthropic funding. As many of you know, the National Institutes of Health and the Department of Veterans Affairs are responsible for the bulk of medical research in our country. And in fact, funding has been going up, which is very much appreciated. And it is the reason that the US is able to be the leader right now in medical research, because of those organizations. However, even with enhanced federal funding, we still need philanthropy. And that's because philanthropists are willing to form organizations that will take risks that these other funding organizations won't. And so they often are willing to take on projects that have a longer time horizon, or that have a lower chance of success, or that require a little bit more input in order to translate it into patient care. And it's very important that we find ways to merge both of these support networks in order to complete this circle of scientific investigation and drug development, where we discover things in the laboratory, we turn it into medicines, those help patients, but then we also learn from the patients, and we go back and learn from them what questions to ask in the laboratory in order to continue improving the field of medicine. And so the last slide, I would just like to thank the major funders for my work. Of course, I'd like to start with the National Institutes of Mental Health and the Department of Veterans Affairs. I would also like to very much thank the One Mind Foundation, which was founded by Garen Staglin, who you'll hear from later today. They supported a lot of my very early discovery work on the P7C3 molecule when other organizations weren't willing to take a chance on that type of screening process. I'd also like to thank the Brockman Foundation, which currently supports an extraordinary level of generosity of support to my lab. And then I'd like to thank the American Heart Association and the Allen Initiative in Brain Health and Cognitive Impairment, which is supporting related projects on brain health, not on targeting neurodegeneration in the way that I showed you with P7C3, but on strategies we're working on in our lab to optimize the functional connection and synergy between the cardiovascular system and the brain in order to keep the brain healthy and functioning better, which we believe will help reduce the incidence of all different types of brain disease. And then lastly, I'd like to thank the Morley family and the Mather family for their support for establishing the endowed chair in neuropsychiatry that brought me to Cleveland.